about the genetic and development of the close cousins of C. elegans, C. inopinata. And with that, I will give you the floor.
All right. So let me just share the screen again.
Yes.
And please let me know if you can see it and hear me.
Yes.
You good?
Perfect.
Okay. And so first, I'd just like to thank you, Adrian, for organizing this thing. It's been really incredible. I'm pretty amazed at the turnout and how the quality of the talks. And also thanks for extending the opportunity to me. I'm really excited to be presenting my stuff with you all. And then, okay, in case you don't know me, I'm Gavin Woodruff. I'm a postdoc in the Patrick Phillips lab at the University of Oregon. I'm going to be talking about C. inopinata. And I'm also, you know, on the one hand, it's kind of sad that we don't get to see the second talk, but it's also maybe exciting for me because I can never decide what I want to talk about. And so I want to talk about all the things that I'm thinking about lately. And also, I'm not one for self-promotion, but I guess it's essential. And anyway, I'm going to be starting my own lab in the fall at the University of Oklahoma. And so I'm really, really excited about that. And so if you're at all interested in this, please send me an email. And then also, you know, always making changes at the last minute. And I just say, you know what, in case everyone leaves, I just want to thank everyone. You know, you don't do this by yourself, right? And so I put the acknowledgements first. And so I must acknowledge that, so C. inopinata is a very exciting species. I didn't discover the species. The species was discovered by Natsumi Gonzaki, who works at the Forestry Institute in Japan. And I was very fortunate to be in his lab when a lot of this stuff was being discovered and found. And I must thank my advisor, Patrick Phillips, who's been very supportive. Eric Hammerschmidt was a former undergraduate in the lab who did all the work on the dauer that I'm going to be talking about. Anastasia assembled the Romanii genome and did evolutionary simulations. John Willis helped with library preps, and a bunch of people provided some interesting feedback on this work. And then I'm going to be talking a lot about the C. inopinata genome. And that was a huge international collaborative effort that was undertaken to assemble this thing. And so my role in that actually was pretty minor. And so their efforts are essential. And so, okay, acknowledgements. All right, let's get into it. And so, yeah, why am I interested in biology? So I think this is interesting. How do different genotypes make different phenotypes? And it turns out that genotypes make phenotypes in multiple different ways. There are sort of proximate within generation causes, developmental, cellular, and molecular processes that allow genotypes to make phenotypes. But then at the same time, genotypes and phenotypes change across generations due to evolutionary forces and also impinged upon by ecological interactions, you know, interactions with the environment and other organisms. And so all of these things are making this happen. So I'm interested in that. And so I think coming from a multidisciplinary point of view, that a satisfying explanation of diversity may require systems where both proximate and ultimate causes can be addressed. And usually after this, I have a spiel like where C. elegans is good for this, and then figs and fig wasps is good for that. But I just want to get into the interesting biology and stuff. And so I'm going to try to minimize the fig and fig wasp and C. elegans stuff, mainly because you guys know all about C. elegans. I don't need to tell you about that. And hopefully I can get into the fig and fig wasp stuff kind of along the way. But the point is, C. inopinata, Caenorhabditis inopinata, is an exciting system for multidisciplinary biology. On the one hand, because it thrives in figs and disperses on fig wasps. And so figs and fig wasps being sort of classic system in ecology and evolution. But then at the same time, it's a close relative of C. elegans, and which is, as you all know, a really great system for molecular genetics, functional genetics, developmental genetics. And so C. inopinata is well positioned to connect these various fields that are really important for understanding how genotypes make phenotypes and how diversity is generated. And so I'm interested in that. So this is exciting for that reason. Here's a sort of summary, a little bit more detail as why it's a really exciting system. And so as I mentioned, it's close relative. Incidentally, I think this is still true. They're discovering species all the time. But at least as far as I know, not only is it a close relative, technically the closest known relative, which is still insane to me, but okay, so that's super cool. So, and we know this is true that a number of the experimental approaches in C. elegans can be extended to C. inopinata. It's super exciting from like an evo-devo perspective. And then at the same time, another reason why it's exciting from an evo-devo perspective is because it looks different. And so I've been working in the world of wormy evolution for a while. And what has long struck me is how similar a lot of them look to one another, even though they're different species, which is, you know, not uncommon, but still, you know, kind of paradoxical. Caenorhabditis is exciting because it's different. It looks different. It's much longer. It develops much more slowly. There's also a bunch of other, a little more cryptic, but still very obvious morphological differences. And so there's all sorts of interesting morphological change that we can ask questions about. And so that's one of the main reasons why I'm interested in the species, to try to figure out the sort of genetic bases of interspecific differences in morphology. And then, as I mentioned, it thrives in figs and fig wasps. It's also ecologically divergent. And so C. elegans doesn't live in figs, or at least fresh figs, as far as I know. We think that most of these Caenorhabditis species are thriving on rotting plants and eating the microbes on rotting plants. Inopinata is found in fresh figs, specifically Ficus septica, one species of fig found in East Asia and the islands of East Asia, like Okinawa, Taiwan, and the Philippines. And so it lives in a really, really different place, which may explain its really different morphology. And it's sort of serendipitously co-occurring with this fig wasp system. And so I'm just going to, I think I said this also to make sure I mentioned Natsumi, and I did. But then also that there's a bunch of work that I've been doing connected to the species that I'm not really going to be talking about because I'm tired of it. I don't want to talk about new things. And so if you're interested in the genome assembly, the species description, and a ton of work, there's this paper. I've done work on the comparative development of body size, like how does body size change within a generation between C. elegans and C. inopinata. I've also done field work trying to understand how C. inopinata lives in nature, like what is it doing? And I'm going to talk a little bit about that to set up the dauer stuff, but there's a lot more there. And so there's that paper. And then also I've measured its developmental rate and its lifespan and these life history, fitness-related traits, which are interesting. And there's that paper too. Okay. And so before I really dig into it, one sort of slice of the ecology that I think is essential for understanding what's going on. And so I say they're associated with figs and fig wasps. I'm going to give a little, I hate talking about it, but I got to give a little spiel about the life cycle of the fig and fig wasp, just because it's unusual. And so, right, so we think of the fig as this fruit that we eat, which it is, but that's the common fig. There's hundreds of species of fig. It's a really speciose group. And the fig is technically not a fruit. The word is ciconia. And what does this mean? It's really an inflorescence with the flowers on the inside. Okay, so it's like an inside-out flower, which kind of doesn't really make sense, because if the flower's on the inside, how does it get pollinated? Well, the fig has a tiny hole just for its pollinating wasp, for the wasp to crawl in and pollinate the tiny, tiny flowers on the inside. And that's what a fig is. And that's where inopinata lives. And so we know it lives on the inside, because if you wash a fig that you pick from a tree, you don't see the worm, but if you cut the fig open, you see the worm. So we know it's on the inside. So it's living on the inside, and the pollinating wasp crawls on the inside. It lays its eggs in some of these ovules while it's doing the pollinating. So that's sort of the mutualism. It lays its eggs in the ovules, but the fig gets pollinated. So we have this mutualism. And so that's that story. They're in this, so these are just pictures from the internet that are really better at illustrating the point than my pictures. These are the actual wasps associated with inopinata. There's two species. There's not just the pollinator. There's also a parasite, and they can exist in the same fig, and they lay their eggs from the outside. And they're also a parasitoid, so they're laying their eggs from the outside into the developing larvae pollinating wasp. And so they're a direct parasite of the wasp and an indirect parasite of the fig, in that they're removing the male function of the fig pollination. And so what I did is I caught these wasps in a bag. So they were in transit, and I put them on plates, and I was like, hey, do I see Caenorhabditis crawl off? Okay, that's one part of the fieldwork that I did. And I only found Caenorhabditis on the pollinating wasp. And so we know they disperse on the pollinating wasp, and they must make some sort of decision within the fig to choose the appropriate dispersing vector. Okay, so that's sort of just one result and an important part of the ecology. Okay, cool. These are the stories. I'm looking at the time. I'm just looking at the clock. There's going to be time. We're going to have fun. Okay, and they're very, very different stories. One very, very simple, probably incomplete, but I like it. Comparative morphology of the dauer in inopinata. The very, very different story on the genome and where the repeats are in the genome. And then super super early results on transcriptomes over developmental time and really exciting for Evo. And now that we're in our surreal pandemic universe, can't really test these things. I can just play with a computer. And so that's what I'm thinking on right now. Okay, so anyway, they dauer. And also, usually, so this is, I'm entering a new phase of the career. Usually I talk about the crap I've been working on. This is not my story. All this work was done by Eric Hammerschmidt, who was an undergraduate in the lab, actually graduated a while ago. He's going to be starting a PhD program at Princeton, I think, which is very exciting. And so anyway, this is his work. Okay, why do we care? And so this is my framing element. I don't know. There's tons of variation in body size in the living world, 21 orders of magnitude. And so what is this? Beautiful illustration from this book here. And within each box, the animals or the organisms are drawn to scale. And the arrows are pointing to the same organism in each box. So for instance, this blue arrow is pointing to a person. Here it's pointing to a person's hand. And I just checked, this is apparently a human liver cell. And so within an organism, you have crazy variation in size, scale of size. And then, of course, among organisms, the variation is tremendous and demands an explanation. Why? What is going on here? And then at the same time, this may be connected to this. Body size has a profound impact, it seems, on an organism's biology, right? And so also forgive the horribly ugly figures. I'm not very good at taking photos, obviously. But the point is the x-axis is the same on all of these. It's body size. And then in this book, they just drew a bunch of things that are correlated with body size, right? So, lifespan, metabolism, various aspects of your metabolism and physiology, a lot of things connected to your ecology, right? Territory or home range, population density. And so it seems like your size is really important for various aspects of your life. Okay. And then we're getting closer to the point, but we're not to the point yet. Here's a neglected but obvious fact of life that I think is interesting and has interesting implications. Basically, that within a generation, if you're like a multicellular developing organism, your body size changes, right? You start small, you get big. Your ecological changes, your ecology also changes as you get bigger, right? And so this is really obvious for things with metamorphism, right? So, you have tadpoles developing into frogs. Tadpoles eat very, very different things from frogs. And so even within a single organism, you have a dynamic ecology, like the things that you're interacting with is changing. This is also true for, say, silkworms, right? And so I think that's what this is. The larva eats leaves, and apparently, at least according to this development textbook, the adult doesn't have mouth, doesn't eat at all. It's just a machine for reproduction. And so different developmental stages have different ecological contexts. And this isn't just true for things with metamorphism. This is also true for things with less obvious, more direct development. And I love this sort of work. It's really cool. And so what is going on here? This is sort of getting at the diet of, I think, largemouth bass as it grows. Okay, and so here we have these different size classes. And the idea is if you're smaller, you're younger. As you get bigger, you're going to be older, right? Or as you get older, you get bigger. And then this is just the content of the stomach. And the young and the small young things eat these like microcrustaceans. As you get bigger, you're eating maybe bigger crustaceans like crawfish and shrimp or something. And then the big mamas eat these big fish. And so different developmental stages. The point is different developmental stages have different sizes. And so they have different ecological contexts. And so how does stage-specific ecology and body size variation influence one another? And so I'm almost to the point. But not only are ecological contexts dynamic due to just the basic fact of growth, many species have specialized developmental stages tied to life history strategies. Okay, and I'm going to get into this thing. This idea of dispersal polyphenism, right? And so oftentimes there are these, especially in invertebrates, these sort of developmental decisions that are really driven by the environment that lead to very vast differences in morphology, just within a single organism, within a single species, right? It's a polyphenism. And so here's a classic example, these solitary and gregarious locusts, right? So if conditions are in I guess, good, you have the solitary morph, and then if conditions are poor or density is high or something like that, you have this drastic change in morphology and behavior. And so this is what causes these crazy plagues, these masses of locusts swarming around. And so that's this, and that this is an environmental polyphenism. And this is another classic example, wingless and winged aphids. If conditions are good, the aphids don't make the wings and they stay in the same place. But if conditions are poor, they have wings, they disperse, okay? And so there's also this other interesting wrinkle in body size development and ecology. And then here I'm getting to the point, Caenorhabditis also has a dispersal polyphenism, okay? And so I love this figure. So it takes the life cycle that all the C. elegans people are very familiar with, and then sort of superimposes it on or puts it alongside what we think these stages are doing in the real world. And as I mentioned, and so C. elegans, most Caenorhabditis species are thought to proliferate on rotting plants. And so this is what they do when conditions are good. When conditions are poor, so, you know, starving, lack of resources, high density, whatever, you have this polyphenism where they develop into this dauer larva morph, where it's specialized for dispersal. They have these specialized behaviors that the non-dauer larvae, like the non-dauer form, they don't do this. The dowers wave their heads on a substrate, and the idea is it's thought this is supposed to aid in hitching a ride on some crawling invertebrate carrier like a snail or a slug. And then that brings that dauer to a new place. And when conditions are good, dauer disembarks and then continues development and reproduces. And so the dauer larva is this sort of specialized stress-resistant stage. You guys know it has all these features that are just really good for dispersal and survival, like a buccal plug to prevent desiccation, presumably, the specialized behaviors, rest of germline, blah, blah, blah. I mentioned the dictation, which is an insane, amazing behavior that we actually are learning more and more about. There's some interesting work that's happening in this area. Okay, the point, variation in invertebrate carrier size. It turns out, at least seems, C. elegans is promiscuous in its invertebrate carrier choice. So it's been found on, so here I got these silhouettes. I went to the literature. These are sort of scaled appropriately. The vectors can be 10 to 65 millimeters long. Caenorhabditis, only found on the fig wasp. Fig wasp is really small, teeny, teeny, tiny wasp. It's not what you think of when you think of a wasp, but it is. And it's, you know, this I pulled from literature, like less than 2 millimeters long. And in fact, that's kind of close to the Caenorhabditis adult size. So what's going on here? The Untitled adult is much longer than the C. elegans adult. What does the Caenorhabditis dauer look like? And so that was sort of the question here. And so we did this, how you did this. And so C. elegans, you expose these dowers to a detergent, SDS, and the idea is that the non-dowers will drink the SDS and get killed, whereas the dowers are sort of not really eating, and so they don't consume the SDS and they live. And that's how we did this. Okay, so going through some figure, I'm writing a paper, so that's what this is. Okay, so this is the thing that we know. Caenorhabditis adults are much larger than C. elegans adults. Okay, that's good. What about the dauer? And so here are the dowers. So the dowers are shorter and wider, and so it's kind of hard to tell from this. They are shorter or smaller in length than C. elegans dowers, but they're definitely wider. That was unexpected. You know, you expect all things being the same, maybe the dowers would be bigger. No, dowers, it seemed like the dowers are a little bit smaller, definitely wider. And so this is something that I was concerned about for a long time, that like, are we actually measuring dauer larvae? And we recently in our lab got a better DIC microscope, and I've convinced myself that this is the dauer because we can see this buccal plug in the inopinata dauer here. And so it seems like this is the dauer. And then I had recently went to Taiwan, I went last year, and got some new lines of C. inopinata. And I was just absolutely blown away when I saw this. And so some of the dowers appear to be ensheathed. And so I don't know if you can see this, but it seems like it has retained the cuticle from the previous molt. And I'm like, is this what I think it is? And I'm like, I don't know if I've ever seen ensheathment before. So I looked at the literature. And so here are some really good images from this paper of, I forget, some sort of parasitic rhabditid. And it's a similar thing. So I'm pretty convinced that that's what this is. And as far as I know, this trait that is the sort of double cuticle thing has never been observed before in Caenorhabditis. Please let me know if you've observed this in Caenorhabditis. I'd be curious to know. But anyway, if not, it seems like a novel trait in this lineage, so another novelty. And this is the big thing. It's usually found in parasitic nematodes. It's oftentimes associated with sort of the infective larvae of parasites. And as an aside, there are other nematodes found in figs and Parasitodipligaster species, which are wasp parasites. They also have this ensheathment, and so this may be something that is really important for the fig wasp lifestyle. Anyway, really exciting observation that I'm interested in pursuing more. Okay, and so anyway, we had some pictures, but can we quantify this stuff? And that's what this is. These are just scatter plots of a simple measurement of width and length in elegans and inopinata, different developmental stages. The gray things are the non-dauer stages, and the red is the dauer stages. And the point is relative to the morphological space of the non-dauer stages, the dowers are occupying this different aspect of the space. So the elegans dower is up here, and the inopinata dower is down here. And so the point is they occupy a different region of morphological space. And this is the 2 dimensions. You can just look at single variate stuff. And so here we're adding a couple more species. You got Briggsii, tropicalis, other hermaphroditic species, and inopinata and elegans. This is length. And so maybe inopinata is not super exceptional with respect to length, although it has the mean that is the smallest of the 4. Its width is definitely a lot bigger, and as a consequence, its length to width ratio is really different. And so they have a pretty different morphologically dauer larva that it is in a direction kind of opposite to what you'd expect given the adult form. Okay. We also were measuring just how often do we see the dauer, and so we did that here. C. elegans and C. Briggsii readily make dowers. C. inopinata really doesn't. They're very, very rare, which is annoying for this sort of biology. And so here I'm showing the same sort of thing, like how frequently do they make the dauer? Basically, the fraction of animals that survive that SDS treatment is what this is. And here, just a bunch of different strains of C. inopinata that I've collected. Here, just from 2 islands in Okinawa, Ishigaki, Itamote, doesn't really matter. The point is that it's not like the colors are all on this side of the graph. They're rank ordered by survival. And it's not like all the red guys you have dauer and all the blue guys you don't. It doesn't seem like there's any association with island of origin and whether or not you have a propensity to make dauer. And a ton of these guys don't make any dowers at all. And the maximum rate of dauer formation is really quite low. And so it's really annoying to study the dauer in this species. Okay, and so that's just that part. That's it. And so really simple comparative morphology with a lot of interesting future directions. And so, right, so they're morphologically divergent from its close relatives and are shorter and fatter, despite the adults' much longer body size. And then this is why I was talking all about that stage-specific ecology stuff. We think, this is my guess, I mean, I don't know, I feel like it should be driven by the natural context of the developmental stages. The inopinata dowers disperse on vectors that are really, really, really small, so the dower should be small. And then we discovered this thing, I think, which is insane. Enchefment seems to be a morphological novelty in this species that could provide a window into the evolution of parasitism. And variation in inopinata dauer formation is consistent with previous observations. C. elegans, right? So like C. elegans, you have a lot of variation, natural variation in dauer formation. Okay, it's also true in inopinata. Okay, so that's the dauer story. Now to something very, very different. Genome and repeats. Okay. Oh, I'm thinking about slides that I don't have in here that I should have, but I can talk about it. You guys can ask me a question later. Anyway, the story's on bioRxiv. And so if you want more information on this stuff, you can read that. And if you're willing to wait, even more information will be available at MBE. So it just got accepted, which is very great. Okay, and so here's framing element. Repetitive elements covary with chromosome position in C. elegans. We've known this a long time. And what are we showing here? So here we're just showing repetitive, this is what I'm calling repetitive genomic landscapes. So here we got your position on the genome, the different facets are the different chromosomes, and this y-axis is the percentage of the genome is repetitive in a 100 kb genomic window. And in all these chromosomes, you got more repeats on chromosome arms, on the ends of chromosomes, and less repeats in chromosome centers. So that's an interesting observation. What is causing this? Why? What's going on? What are the patterns? How do they vary? It's interesting. And so, yeah, so we live in this amazing time where not only do we have all tons of genomes, we're actually getting better quality genomes. And so we can actually look at stuff and how this varies between species. And so we looked at the repetitive genomic landscape in 5-worm species to understand how they evolve. And so these are the assemblies that we used. They had pretty good chromosome level contiguity. And these are those citations if you're interested in that. Oh yeah, so right. So here's something that I didn't mention because I'm not really focusing on it. But in case you didn't know, so there's like, to me, there's like the obvious thing in Caenorhabditis variation is reproductive mode. And so C. elegans is famous for being a selfer. It turns out most species in Caenorhabditis are not selfers. Most are male-female, like us. And C. inopinata is male-female. And so in this little panel, we got 3 male-female species, Inopinata romanii nagoni, and 2 selfing species, Briggsii and elegans. Okay. trying to be brief on the methods. The point is that we took the assemblies, just the FASTA files, and used the same pipeline to annotate repeats for all those assemblies. And we used a variety of methods, both these sort of de novo inference methods, repeat modeler, and then also these methods that sort of use a database of known repetitive elements of a certain type. And so we combined a bunch of those to get the repetitive genomic landscapes. Okay, and that's what this is. And so before I just showed you 100 kb windows and elegans, now I'm showing 10 kb windows, so more granularity, and 4 of the species. And so 4 of the species share a conserved non-random genomic distribution of repetitive elements in all the chromosomes. The same thing as before, you have more repeats on chromosome arms and in chromosome centers. However, Caenorhabditis is very different. So it does not reveal this genomic organization of repetitive elements. It seems to be pretty uniform in its repetitive genomic landscape. And so not only is it different in its ecology and its morphology, it's very different in its genome, at least in its structure like this. Okay, and so that's an amazing observation, but can we dig into this? And so what about different kinds of repetitive elements? What do the repetitive genomic landscapes look like? So it turns out there's this whole menagerie and taxonomy of repetitive elements that have very, very different sort of sequence signatures and modes of replication. And the point is, just from this figure, is that different kinds of repetitive elements have different genomic landscapes. And so here I'm just showing a few repeat superfamilies, and some of them have conserved landscapes. Maybe they differ in abundance. Here's one that both in inopinata and elegans have that sort of familiar center arm shape, but some are more uniform. And then you have differences among species, right? And then you have some that are really quite different, like say TC1 mariner, where they appear to be enriched in chromosome centers in inopinata, whereas they're enriched in chromosome arms in elegans, or RTE, which is almost non-existent in all the species, but in inopinata, where it's gone crazy and it's highly abundant. And you could do this at all the species. And so there's tremendous diversity, both in repeat type and repeat distribution across the genome. And so that's fun to look at. We wanted to put a number on this thing so we could dig into it a little bit more. And so how did we do this? So this is a little complicated. So I'll just try to hopefully explain it as best I can. But the idea is we decided to normalize by chromosome position for all the chromosomes. And so sort of set the center of the chromosome to 0 and the edge of the chromosome to 0.5. And so this is something like, imagine all the chromosomes folded in on themselves, like the folded repetitive genomic landscape. And then what you can do is like, okay, we have this sort of folded repetitive genomic landscape. If it's less than 0.25, we're gonna call you a center. And if it's greater than that, we're gonna call you an arm. And we do the arms minus the centers, we get a number as to like the repetitive genomic landscape. And to sort of maybe make that more clear here, technically this is the arm center difference Cohen's d effect size of repeat density in the arms and centers. But the idea is say if that number is negative, you got more repeats in the centers. If it's positive, you got more repeats in the arms. And if it's close to 0, there's no difference between arms and centers with respect to repeat density. I'm skipping around here, but so now that we have that number, now we can check that number with a repeat superfamily. And that's what we've got here. And so here, just looking at Caenorhabditis, rank ordered by abundance in the genome, the various different the most abundant repeat superfamilies, and colored by that arm center difference, or this thing that I mentioned before. And so if it's red, it means it has that sort of M shape with the more repeats in the arms, whereas if it's more blue, you have more repeats in the centers in the arms. And the point is the most abundant repeat superfamilies in Caenorhabditis appear to have atypical genomic organization, right? They aren't red. So if you remove those, turns out Caenorhabditis underneath those crazy superfamilies is a more conventional, familiar repetitive genomic landscape. So it seems like these superfamilies of very, very different replication modes, by the way. So you have DNA transposon, line retrotransposon, LCR retrotransposon. So very different types of repeats are driving this very different repetitive genomic landscape in Caenorhabditis. Okay, but what's going on with these families? Like, what is driving this? And so I'm going to talk about 2 ideas as to what may be driving this. I don't think I've really decided as to what it is. It's probably multiple things that are causing repetitive genomic landscapes, but I like this idea, so we'll talk about it. There's another pattern. So it turns out all these genomic features are covariant with each other in the worm genomes. And one of them is gene content. And so before I was looking at repeats, now we're looking at genes. And in C. elegans, there are more genes in chromosome centers than in arms. And so maybe that's causing it. And so you could imagine if a repeat wants to land in the chromosome center, that'll be pretty deleterious because there's lots of genes there. So maybe it wants to land in a place where there's less genes, right? And so if this were true in an inopinata, okay, wait, sorry. So if this is true, I'm gonna have a different way of showing this data. So the idea is if this pattern holds, you'd expect a negative relationship between gene density and repeat density, right? Because genes are high in centers and repeats are low in centers and vice versa in the arms. And that's what I'm showing here. And so here, just plotting gene density and repeat density for all the species. And in most of the species, see a negative relationship, like you'd expect, but in inopinata, very surprising, you see the opposite pattern, a positive correlation. And it's like, what? This doesn't make sense at all. And so previous analyses, and so I've been playing a lot in a computer, and I noticed this, and also actually this was also incidentally published in the genome paper. It's in one of those big supplemental tables. But previous analyses revealed Caenorhabditis protein coding genes have a number of transposon domains, or a number of them have it. And so I just wanted to do this with all the Caenorhabditis protein sets. So before, I was looking at just 5 species because only 5 species have really good chromosome level contiguity. Now I'm getting all the protein sets that I could find, and that's here. And what are we doing? We align them to a transposon database. So you take all the protein coding genes, or all the proteins, and you align them to a transposon database. And here it's just measuring what percentage of those proteins align to the transposons. And so for most of the species, including, say, C. elegans here, like less than a percent of their genes are aligning transposons. However, C. inopinata is weird. Also, like C. japonica, apparently, in that tons of its predicted genes are aligning to transposons. More than 15%. That seems like a lot to me. And so I remove those. And if you remove those, this correlation goes away. There's no gene repeat relationship in C. inopinata. And then I'm going to show another way of looking at the genic landscape in the genome. And so here, again, we have gene density and the different species, inopinata, and then inopinata when I've removed those genes that align to transposons. And here we've just binned by chromosome center and arm. And so in C. elegans and in most of the other species, as said, you have more genes in chromosome centers than in arms. But in C. inopinata, if you remove the genes that align to transposons, seems to be no difference, or a very minor difference in the enrichment of genes in centers relative to arms. So it seems like the C. inopinata has lost an ancient organization of its genome, both in repeats and genes. And so that may explain, so having that loss of gene structure may explain its repetitive landscape, but hold on, combination rate also co-varies with everything. And so there's just all this stuff co-varies with each other, so it's really hard to disentangle cause and effect. And so what do I mean by this? And so here are some Mary maps showing, so it basically plots your sort of physical base pair position with your genetic sort of, Morgan position recombination. And the idea is these gray lines are showing the slope of those things, which is telling you something about the recombination rate. And the point is that recombination is higher in chromosome arms and lower in chromosome centers. And this is true for all the chromosomes. This is also true in C. briggsii. It's probably true in a lot of Caenorhabditis species. And so ideally what you'd want to do is measure recombination in inopinata, which I'm going to do. Haven't done yet, but we could do something else. Can recombination alone explain distributions of repetitive elements? And we used evolutionary simulations to get at this. There's this amazing sort of language software package, Slim, that allows you to simulate rate fisher populations. And there is even a recipe within that manual for copy and paste TE evolution. So we just grabbed that and added some things that we wanted to test. And so really we simulated a 3 megabase chromosome, and this is the key thing. We compared basically a chromosome with uniform recombination along that chromosome. And we simulated a chromosome, a set of scenarios with 3 domains of recombination domains, like the worms. So with high, low, and high recombination. And then various fitness effects of insertions. And so I'll talk about that. Hopefully that'll make sense. And so these are the results here. And so here it's the same thing. It's going to be the repetitive genomic landscape, but this time not a worm, it's in a computer. And here we have the repeat density and the position. And then this facet is going to be the uniform recombination. So there's no differences in a recombination rate along that chromosome. And then here it's going to be high, low, and high recombination. And then if it's red, it means that, yes, there's a repetitive genomic landscape between the different domains. If it's black, it's pretty uniform repetitive genomic landscape. And then the darker shades of gray in the background are corresponding to more deleterious effects of insertion. So we're also going to test at the same time if you're more deleterious when you insert in the center, which would you'd expect to maybe be true if you have more genes in the center. Like, okay, so that's what we're kind of getting at here. Okay, so if you have this sort of uniformly neutral, no difference of fitness insertion effect, whether if it's neutral or highly deleterious, regardless of the recombination domains, you don't see any repetitive landscapes. However, if GEs are uniformly weak in their fitness effect, and you have these domains of recombination, you can see these repetitive genomic landscapes. However, if you have these sort of different domains of fitness insertion effects, so if it's more deleterious in the center than in the arms, you also see repetitive genomic landscapes, even when there aren't domains of recombination. And so in a variety of scenarios, genomic heterogeneity and fitness insertion effects or insertion fitness effects are sufficient for generating repetitive landscapes. Okay, and so that's sort of the end of that part. And so Caenorhabditis has a divergent repetitive genomic landscape. It's really crazy. This pattern is largely driven by 4 divergent transposable element superfamilies. Caenorhabditis has lost an ancestral genomic organization of protein coding genes. And genomic clusters of insertion fitness effects are sufficient for generating repetitive landscapes, although domains of varying recombination can cause such landscapes when selection is weak. And relaxed selection on certain repeat types due to the loss of genetic clusters could explain the genomic repetitive landscape of Caenorhabditis, but other historical or genomic factors are needed to explain this pattern because your repetitive landscape depends on what repeat you are. And so there's more work to do in this area. And then I just must add, because it's like the smoking gun for the repetitive elements, Caenorhabditis has lost, I think, Ergo1, ARI7-9, and so it's lost a couple of these RNAi pathway genes, which is sort of like, oh, maybe that's why. It has this crazy repetitive genomic landscape, and I'm very keen on following up on that as well. I didn't include that slide here. Okay, so that's that story. That's the genome. That's the repeats, which I think is super cool and crazy, and very surprising and interesting. And then now, the new stuff, really, I put it, great. So are you ready for some candidates? What is this? And so getting into the transcriptomics of body size evolution. And I have time, we can talk about this. This is gonna be cool. And so this was the obvious immediate thing about this species that makes it exciting for Evo-Devo. It's different, and it's different in its size. Why? I'm interested in both reasons why, proximate and ultimate reasons, but we're gonna talk about some proximate things today. So what are the developmental and genetic changes that are driving this difference? And so I'm going to talk about some of those obvious candidates in the future slides, but for right now. And so there's a talk a couple weeks ago, I think it was Kathy Savage-Dunn gave a great talk on body size TGF-beta stuff in the virtual worm session. And something that is true about those mutants is also true about Caenorhabditis, in that the body length is not always big over time, right? And so here we're just comparing length over developmental time. When you synchronize them at the same time, this comparison is not super great because Caenorhabditis developed slowly. Nonetheless, earlier in the day, and also this is at 25, I feel like because you're worm people, I have to say that. This is at 25 degrees. Early on, they track each other pretty well in length, and it's really at the larval to adult transition that Caenorhabditis gets much bigger than C. elegans, okay? And so what are the patterns of gene expression through this developmental period, and how do they change between species? And could these differences be important for the evolution of body size? And so that's what we're getting at. Like what are the gene expression patterns going on through this developmental window between these species? And so we did the RNA-seq to get at this. Very simple comparative RNA-seq thing. And so we did 30 samples. We did the 3 stages. So sort of before, during, and after that body size difference becomes more apparent. The 2 species, 5 replicates per group. Great. I usually haven't talked about this in other talks, but I feel like for the worm people, I must share. I used the FOG2 mutant in C. elegans, and so FOG2 is important for hermaphrodite-specific spermatogenesis, but not male-specific spermatogenesis, and so it's like a male-female strain. And so I didn't want my results to be driven by the presence of males, and so that's why I did this. And I didn't want to pick males and females because I'm lazy. Maybe I'll do that later. We lost some samples for reasons you lose samples. Also, this is a tricky experiment, and it's also why I want to do it again, because you cannot isolate these stages at the same time, because Caenorhabditis develops so much more slowly than C. elegans does. And so they weren't isolated at the exact same time. So either you have to you have to synchronize them at different times and then isolate them at the same time, or you have to synchronize them at the same time and then isolate them at different times. And so I did the latter. And so I'd really be curious to know what these results would look like if I did them again. Okay, so the workflow, it's already seek. And what are the major things? Major things is, right, we're looking at about 10,000, 11,000 single copy orthologs. And so unfortunately, you got to throw away a lot of the genes when you're comparing species because of, copy number variation and gene loss and stuff. Yeah, we did the analysis in R and all this stuff. And I love the worm-based tools. They're super fun. Okay. And so I used to have more slides about this, but it's like, Gavin, you're going on forever. No one cares about this stuff. And so we did the gut check data exploration things. So like PCA. And so the point is if you're the same color and the same shape, that sample is from the same group. And the point is that organisms with the same group cluster in multivariate transcript space. And what's nice is that this first principal component, it's separating the adults from everybody else. And the second component is separating the L3s and the L4s. It's like, yes, seems like it's working. Okay. And then another way to gut check, is this working? And so we just plot sort of C. elegans expression and an inopinata expression. This is within L3s. This is the mean expression for a given gene. And gene expression levels are broadly correlated between the species. So that's also like, okay, seems like it's working, probably working. It's like, okay, cool. Seems like the RNA-seq works. And so this is the big question. Within a stage, how many genes exhibit significant differences in gene expression? Turns out, gene expression differences all over the place. So these are volcano plots. Red means the p-value is, what is it? Adjusted p-value. This is BH adjusted p less than 0.05, so significant. It's all over the place. And also, so these volcano plots, if this is a full change, so it's greater than 0, you're more expressed in an inopinata, less than 0, more expressed in C. elegans. And then this is the negative log of the p-value. And the point is, it's just in the L3, L4, and adult, the vast majority of genes are differentially expressed. In adults is like 66%. So just lots of, yeah, okay, that's right. Yeah, just tons of differential expression. Okay, so cool, most genes are differentially expressed. Now what? And so we're getting close to the end. I'm just gonna talk about some of the things that I'm thinking about as far as this data. And, because you got so much variation in gene expression, it's very fun to look at the lists and to fantasize about candidates. And so that's what this is going to be. But I think there are some satisfying, interesting things here. So let's just go along for the ride. Okay. So now what? Look at some candidate genes from background literature. Okay. And so before there was even a C. inopinata genome assembly, I had the vision in my head of what the genes were that was going to be causing the body size difference, because we have all these mutants from C. elegans that have differences in body size. And so, so this DBL1, it's the ligand, BMP basically. If you knock it down, the worm is small. If it's dose dependent, if you add more, apparently it gets longer. And I thought it was like, oh man, there's copy number variation. This thing is duplicated in an inopinata, and that's why it's long. No, there's only one double one in an inopinata. Okay, what about gene expression? Like maybe it's not copy number. Maybe they're differentially expressed. And so here are some simple hypotheses that we can ask right away. This experiment. I think this is great. Finally, asking some genetic hypotheses. Okay, so one hypothesis I just mentioned. Well, maybe this thing is overexpressed in inopinata. That would be cool. But then there's also, and then like this is classic, right? Like this is one of the first papers I ever read in C. elegans, and it was one of the striking genes, right? You knock down a gene, and it gets longer. Like, oh, inopinata has lost this LON1 or LON2. No, inopinata has all those genes. In fact, not just the named LONs, it has all of the, at least last I checked, all of the genes that have a long phenotype in worm-based C. inopinata has. It's frustrating. Anyway, so, but maybe they're not, maybe they're underexpressed. Like maybe their expression is lower and that's why it's longer. Okay, those are the simple hypotheses. And so, right, so just looking at those TGF-beta pathway genes. Oh yeah, I guess I just failed to say, like TGF-beta pathway seems to be the important pathway for regulating body size. And so let's look at those genes that are connected to this pathway. Sorry, obvious part. And so that's what these genes are. I just took this from this review, and we're going to look at their expression. And this is, again, that log fold change. So if it's greater than 0, it's going to be more expressed in inopinata. If it's less than 0, it's going to be more expressed in C. elegans. And then we have the stage. So those are the 3 stages. And then we have this, other feature. Basically, you want to know, like, is the P, is it significant, like how, some significance threshold. And so if it has this black box. It passes that threshold. And the point is that there's variation all over the place. Most TGF beta signaling genes are differentially expressed. And so it's like, whoa, that's amazing. But what about our hypotheses? And so remember our hypotheses about this gene? The hypothesis is that it would be overexpressed in inopinata. No, it's the opposite. It has lower expression in C. inopinata, the opposite of expectations. Same can be said for these long genes. These long genes have high, if at all, so some stages there's no significant differences in expression. In the stages where there are significant differences in expression, they have higher expression. And so it's like, okay, that's cool, opposite of expectations. And then you have a number of genes which have these intriguing patterns where the dynamics seem to be changing, or rather the difference in expression is increasing or decreasing as the worm gets older. So that's super interesting too. So we got some interesting things there. So the point is that most of these things are differentially expressed, and you have sort of these inscrutable equivocal patterns. Maybe you got some developmental system drift. And so we will be following up on that later. I think it's super interesting, but sort of hard to interpret, right? Okay, and so we did that, but hey, how about actually doing some statistics? And so we can do that too. So this was the key thing. Key thing is that, can we get genes whose expression dynamics across development are different between the species? And so the body size is getting bigger as the animals, as both species are developing. What about the gene expression differences? Can we get genes where the dynamics are changing across development? And so the way we do this is with a model comparison approach. Basically, so yeah, I realize I'm going way long, and so I'm just going to kind of speed up. But the point is we want those genes that maybe have different dynamics, that have a species and time interaction. And that's what we're looking for. And so we can compare those two models. So either basically they have the same slope, or they have different slopes. And so is this one more likely than this one? And so that's what we did, likelihood ratio tests. And there are so many genes, again, tons of genes that are significant with this approach. And this is adjusted P, like it's crazy. And so I just said, okay, so for my list for ontology enrichment and things like that, I'll just take the top 10% by p-value. So are you all ready for some candidates? This is number one, exactly what we're looking for. It's going up, and so this is the same thing as those toy models I showed before. So we got stage, got expression. Going up in inopinata, going down in elegans. So great, this is number one. We got the things we wanted. Turns out this is ACE3, an acetylcholinesterase, which is interesting. And then we got some other genes. Lin31 is exciting, has different dynamics again, because it's implicated in TGF beta signaling. Lin42 is exciting, it has different dynamics, seems to have maybe constant expression in inopinata where it's going down. In elegans, this is exciting because it's implicated in developmental timing. It's like a heterochronic gene. If you knock it down, you have slower development and more variable development, and this is also true in inopinata, so it's exciting for sort of maybe getting at the evolution of heterochrony. We have some body size candidates in this list. ZIP10, if you knock it down, the worm is longer, seems to be regulated by SMA9. In addition, RUNT1, if you knock it down, seems to be smaller. And then they have sort of similar dynamics here, ZIP10 and RUNT1, maybe constant, maybe going up a little bit in inopinata where it's going down in elegans. And then these two genes are also exciting because they were the top body size targets of a SMA9 mutant microarray experiment. It's like, wow, that is amazing. And then also, this is also sort of, and this is just, speculation, my excitement. But it turns out a number of years ago, I did a molecular evolution study looking at models of codon evolution, signatures of lineage-specific selection in inopinata. I looked at about 2,000 genes. Only 20 genes had significant evidence of this lineage-specific selection, and one of them was SMA9. And so you have maybe a gene that appears to be under lineage-specific selection, and then its targets are changing their dynamics potentially. And so this is sort of very speculative, but there's some exciting things going on here, some good hypotheses. But okay, what about the gene ontology? Please forgive me if I'm going way too fast now. I'm just gonna maybe just summarize this stuff. And so, so worms are cool because they have species-specific ontologies. It's not just boring old Go. You can look at anatomy terms, so the genes that are on my list with different dynamics between elegans and inopinata, where are they expressed? It seems to be neurons. Neurons is the big thing. You saw ACE1 being that number one gene, and so the arrows are pointing at sensory or motor neurons. The only thing that's not a neuron is a socket cell, which is pretty close to a neuron. And so the top anatomy terms are dominated by neurons. The phenotype terms, of course, because of the neuron thing, they're largely behavioral. You got this behavior variant, copulation variant. This may be because of the male-female thing. But also notably, we got this body morphology variant, and so that's exciting. There's some body size things in there. For conventional Go, the top thing is molting cycle, which is cool because, as I said, the inopinata develops twice as slowly. C. elegans, and so maybe there's differences in the machinery that guides cyclical events like molting. And so, okay, so how does all of this compare with previous elegansomics experiments? And this, I must thank the Virtual Worm Club, because I didn't know about this tool. I'm like, I want to use that. and this worm X thing that I think, I forget who did it, but in one of the earlier sessions, this was shared. And I'm like, I want to use this. How did I not know about this? And what it does is it has a list of lists. And so I'll just read it. We collated all published high throughput expression data sets of C. elegans. And it offers tools for performing enrichment analysis based on the taxon-specific, now 2,000 specific gene lists. And so it compares my list of things that have significant differences in expression dynamics over developmental time and compares it to lists from all these C. elegans studies. And get this, I was totally surprised, always surprised. Like, number one, and so what is this? So these are the lists and the percentage of my genes that overlap with that list. And these are like the most significant. They're like with the lowest p-values. These are the top 20 with the lowest p-values. And number one is DAF2 versus wild type. So you got a number of things that include the dauer-related stress response. I suspect my worms are really stressed out. I think there's some other anecdotal evidence that might suggest that that's right. Also developmental timing related, kind of probably what's also driving that molting thing in the go. And then this also is insane to me. Small RNA heterochromatin related. So we got, I think, ALG1 or AIN1 or AIN2 is an argonaut, and then one of their cofactors. And so you got these argonaut related things, small RNAs, and then also heterochromatin things. And this is pretty cool because of a talk on this gene that was at the TGAC. And it's like, oh my God, why? Because could this have something to do with Caenorhabditis genomic landscape or repetitive elements, right? So we know that small RNAs and heterochromatin is somehow implicated in the control of transposable elements. And so I'm interested in looking at that as well. And so, okay, that's the concluding parts of part 3. And so the vast majority of genes are differentially expressed across all developmental stages, including a number of these TGF-beta pathway components. Moreover, the vast majority of genes, not only are they just different within a stage, their dynamics across the stages are different. And genes with more dissimilar dynamics tend to be neuronal, but they also include genes important for molting and body morphology. And then are also, of course, you can pick your favorite candidates. And then the comparison of the above genes with previous elegans experiments reveals overlap with stress response, developmental timing, and small RNA chromatin regulation. And of course, I want to keep pushing on this. There's not just those ontologies that I looked at. There's a bunch of different ones that I would like to play with. I want to look at more than just the one-to-one orthologs. I want to look at how these transposons are expressed. I have looked at this a little bit, so if you have questions about it, I can do a spiel on that. And then, of course, I want to actually, put my money where my mouth is and do some functional genetics, because I haven't really been doing that. And that's going to be essential for really harnessing the promise of this system. And then, of course, I want to do it again, because I suspect I would get different results. Or I don't know. I'd like to try it again. And then that's not all I'm working on. I'm working on all sorts of stuff. Of course, interested in Evo, Devo, and genomic organization, and really pursuing these hard. I also am working on the genetics of variation, population genomics, life history evolution. We have all of these amazing comparative genomic tools, and I want to put them in a phylogenetic framework. And then, I know a lot of y'all are interested in sort of the wild microbes. I'm interested in that too. I want to do sort of the 16S sequencing. I also have a number of wild microbial isolates that I've gotten from the field, and I want to see how they interact with my worms. I did the acknowledgements first. Thank you for listening.
All right. Thank you, Gavin, for that very exhaustive information. We already have like a few questions in the chat. Again, if you want to ask a question with your mic, you can virtually raise your hand on Zoom if you click on the participant button on the bottom, and then on more, you can raise your hand, and then I'll unmute you, and then you can ask a question. Otherwise, you can write your question in the chat. So the first question was from Etenu. So given their association with rotting fruits, do they still grow well on OP50?
Yeah, so they do grow on OP50, but I would say not well. Their fecundity is much, much lower on OP50. So I say it's much, much lower. So the question is, they have a low brood size in the lab. Is that because of the environmental conditions, or do they actually have that brood size in nature? And so I'm assuming it's probably the environment. And so they have low brood sizes, like 50 to 100, even in the presence of continuous mating. Or wait, you know what? So it also depends on, they're sperm limited too. They have huge sperm. And so if you only let them mate overnight, they have lower brood sizes, obviously. But if you let them continuously mate, it's still pretty low, 100, 100, so pretty low brood sizes. Also, their germlines are really wimpy, at least compared to C. elegans. And so in fact, so this was a result that I had, is that they have about the same amount of somatic cells, C. inopinata and C. elegans. But if you counted the germline, C. elegans would have far more cells because their germlines are big and healthy. The inopinata germline is really small. And so I think they really hate the lab. And so they do grow, but they grow slowly and they grow. And it's also unclear if the slow growth is like a fundamental component of their biology because of their Fig context, or are they growing slow because the lab sucks as the environment for them. Notably, there's another Fig species, species 35, and I've lost it multiple times in culture. And so some of these Fig species you just can't grow. And so that's another piece of evidence that the environment's pretty bad.
That's what they feed on in the Fig?
I'm sorry?
Do you know what would they feed on inside the Fig?
That is really what I'm curious about. Like, that's why I want to do the 16S stuff. So
right.
They can eat E. coli. Presumably they eat bacteria.
Mm-hmm.
What are the bacteria in the figs? Yeah, I really want to know this. Yeah.
All right. Okay, I'm going to move to the next question from Peng A. I am wondering how many transposon genes are in C. inopinata. When those transposon are removed, does it reduce the overall gene counts?
Mm-hmm. And so, yeah, this is the thing. Is it a gene if it's expressed? And so this is the thing that I'm really curious about. And so, yes, if you just say, if you align to a transposon, we're going to remove you. Yeah, they have something like 17,000 genes. So they have less genes if you remove them. But the question is, should we? And so why were they annotated that way? Presumably because they're expressed at some point. And so, yes, if you remove them, inopinata has less genes, but maybe they're genes. So yeah, this is what, does that make sense in my answer? And so I don't know if they should be removed. And so I still want to dig into their expression and those sorts of things. But yeah, like that's super interesting. I think they have less genes probably, yeah.
Another question from Peng. He's wondering how long does it take for the inopinata to surge in the body length? And did you see any particular gene expression jumped with that time window? Or
yeah, and so that's what I was trying to get at with the differences in expression dynamics, because you could imagine it could go both ways. Like maybe there's negative regulation, and so it's the spike has to go down, or maybe the spike has to go up, or maybe it's the dynamics, like it has to be on all the time, whereas in elegans it's going up and down or something. And so you could isolate genes in that way, like you could get some number. I haven't looked at that in particular, but that's another way to do it too. It's like I have really specific expectations for what the dynamics of this gene should look like, and I can capture those too. So yeah, that's a good idea.
Yeah. Next question from Louis-Mathieu Harvey. About the genomic landscape of the repetitive elements, have you investigated the possible difference in the regulation of this transposon between C. inopinata and C. elegans? Are they more allowed to be expressed?
I mean, yeah, this is exactly what I'm interested in going forward. As I mentioned before, I should have put a slide on it because it's just such an obvious fact that C. inopinata, I think, so I know ergo1 is one RNAi-related gene, pioneer-related gene that inopinata has lost. Also, I'm not an expert in this stuff, so I may be totally wrong, but I know it's lost ergo1 and I think ARI7-9. And so those are RNAi adjacent genes. And so that might be why its repetitive landscape has gone crazy. Also, with respect to the sort of genes with different dynamics, there seems to be overlap with these ALK2 AIN1 mutants. And so maybe their dysregulation or change in regulation is important for a repetitive genomic landscape. I didn't mention it because there's just a million candidates, because this is another thing, like if there's a million candidates, you can get a million stories. One of those genes on the list is PRG1, which is really important for pyrRNA regulation, which has been shown to be connected to transposable elements. So yeah, I'm really keen on investigating what the hell is going on there, what specific genes are involved, yeah.
All right, next question from Katie Savage-Dunn. Is there any differences, any evidence for change in lipid accumulation in C. inopinata?
I have no idea. I suspect their metabolism is very different, but I would be interested in that. But yeah, I don't know.
All right. Next question from Michael Alien. Were the dauer just induced by starvation? Could inopinata dauer be induced by something different, like wasp smell?
Yep, I agree that is probably what's happening and why we see so few dauers. Like both for dauer entry and exit, you probably need fig and or wasp or both. I've tried growing these things on fig plates from figs you can get at the supermarket. They hate it more than the regular lab conditions. I need to figure out how to culture Ficus septica or just do something with the figs themselves, because I think it would be really interesting and for their biology. Yeah, but yeah, I agree. Like really dig into the dauer stuff. You want to get some Fig and Wasp stuff. Yep.
Two questions from Jonathan Eubank. First question, is there any indication that polyploidy is the driver for body size as in C. elegans? And question two, you only looked at orthologous genes. Does the Caenorhabditis specific gene show similar overall pattern?
Okay, so two big questions that I think I can answer. I personally don't think polyploidy is driving the difference. And so we did this work in my Evolution Letters comparative development paper. And so germline ploidy is the same. So they have 6 chromosomes. We know this both from the genome assembly and from, cytology, karyotyping of oocytes. And then also I looked, so there's also this, well-known somatic polyploidization. So the hypodermis undergoes sort of nuclear division without cell division. And that's thought to be important for body size change. I looked at that using confocal microscopy. I didn't see any increases in somatic ploidy, either in the intestine or in the hypodermis. I still don't, how do I say this? I didn't see anything. I'm just gonna say that. I don't think polyploidy is driving it. And then orthology. Oh yeah, so one of the big things, oh my gosh, there's probably all sorts of differences that I should know that I'm not aware of, but one obvious thing is odorant receptors. And so inopinata has far fewer odorant receptors than C. elegans does. And so C. elegans, I think odorant receptors is like the biggest gene family in the species, something like 1,000 to 2,000 odorant receptors. Inopinata has far less, like hundreds. And so, and we think that's probably because of its ecological context. And so it doesn't, elegans is found all over the world on all sorts of substrates. Inopinata is just found in this one species of fig. And so it probably doesn't need to smell as many things. That's my speculative guess. Yeah.
Okay, we're going to go for the last question for the sake of time. And the follow-up question can be asked on the Slack channel. So last question is like, regarding the ensheetment, which is a cool result, do you observe tube nictation just like with the unsheeted dauers of the Aonema genus nematode?
Man, I should know what tube nictation is because I've read all this Aonema stuff. Aonema is an awesome species that hurts my brain. I don't know, and so I know they do nictation. I don't know about I don't know about tube nictation, and so I think that's maybe they, what is it? They use their own cuticle as a substrate or something. I don't know, and so yeah. So right. So does ensheathment bring with it a bunch of new sets of novelty of novel behaviors that it can do now that it has this sheath? That would be really cool to look into. Yeah. And then the follow-up, does that mean that the underlying mechanism, I'm assuming the underlying mechanism driving body size is different from C. elegans? I would say that among C. elegans mutants, there's a ton of different mutants, and I suspect there's many different ways you can make a worm bigger, and it doesn't necessarily have to be polyploidy, but I don't know. Yeah.
All right. Well, thank you very much for the talk, and thank you for everyone for being there. We'll see you next week with some more development talk and big data.
All right.
All right. Thank you very much. Have a
Thanks.
nice week and stay safe.